Okay, so for the today's class, the next topic is gram staining. First, I will explain what is gram staining and why it is required. Okay, so there was a scientist named Christian Gram. He saw that there are two groups of bacteria. The first group of bacteria, which has a different cell envelope structure. We are done with cell envelope. Now, what we are studying is grouping bacteria based on the structure of cell envelope. Okay. Now, there was two groups of bacteria based on the cell envelope structure. He saw it like about morphological structure, that he saw that there are two group of bacteria. If I take a bacterial inoculum, you all know about what is bacterial inoculum. You all know what is bacterial inoculum. Okay, I will explain. Bacterial inoculum is if I take a sample of bacteria from soil, I put it in a nutrient media and then allow it to grow within that nutrient media. That nutrient media now become an inoculum for that bacteria. So inoculum becomes a sample of bacteria. Clear? We can make this sample for other bacteria also, other organisms also. But for now, I'm taking a bacterial inoculum. When he saw that when he is taking a bacterial inoculum on a slide, on a glass slide, then he can see two different structure of cell envelope of bacteria. Now, how he will classify these bacteria, and then he found out about the gram staining. So gram staining is named on his name, Christian Gram. Now, what is gram staining? If anyone on the online class, if you cannot understand, you can raise your hand and ask. Okay. Gram staining. When he took a glass slide, and on that glass slide, he put the bacterial inoculum. So number of bacteria was there on that glass slide. Okay. Now, when we leave the glass slide to dry, then bacteria get attached to that glass slide. Now I will put the first stain, that is crystal violet. What is crystal violet? Crystal violet is just a stain. You have heard about safranin, you have heard about cotton blue, fast green. These all are stains which are used for different organisms. Okay, so when I take crystal violet, and that crystal violet helps the bacteria to stain violet. This stain was used by Christian Gram, so it was also called as gram stain. Understood? Understood. Now, after this, all bacteria was violet in color. Then what he did, he put Ki, potassium iodide. What is potassium iodide? Potassium iodide is mordant. Mordant is a chemical which helps in making the bond stronger between the pigment and the structure we are staining. Understood? Here we are staining the bacterial cell envelope. And if we are staining bacterial cell envelope with a stain, so what will Ki will do? Ki will make the bond stronger between the cell envelope and the pigment. Understood? Understood? Pigment means stain. Clear? Clear. Next point. After this mordant, he used alcohol wash. After alcohol wash, when he again saw the bacteria under the microscope, he saw that there are some bacteria which lacks pigment and which have pigment. Those bacteria which lack pigment, he classified them as gram-negative bacteria. And those bacteria which have pigment, he classified them as gram-positive bacteria. Okay. Now, what was the basic of this classification? When he took two of the bacteria, which was gram-positive and gram-negative, then he saw that the gram-positive bacteria has plasma membrane, it has cell wall, and it has glycocalyx, but no other layer over the glycocalyx. But when he saw gram-negative bacteria, he saw that it has plasma membrane, it has cell wall with glycocalyx, and over glycocalyx, there was one more layer of lipopolysaccharide. Okay, lipopolysaccharide means?
Say that again?
Lipopolysaccharide. Okay.
What did you add lipopolysaccharide to?
Lipopolysaccharide. Lipopolysaccharide means when lipid and carbohydrate forms a structure. That's the basic meaning of that. Okay. Now, lipopolysaccharide, as it is a lipid layer, then it will be washed off due to alcohol. That we know. Okay. When it is washed off due to alcohol, then this gram-negative bacteria loses the color. Why? Because when the stain was present, then the stain only come till this lipopolysaccharide layer, not beyond that, because there is a thick lipopolysaccharide layer. On the other hand, in this gram-positive bacteria, the stain was come till this cell wall. Understood? Understood this? Okay. Now, in gram-positive, we saw the stain, and in gram-negative, we didn't saw the stain. Why? Because gram-negative was having an extra lipid layer, which was washed off due to alcohol. So he concluded that gram-positive are those bacteria which don't have any extra layer over their glycocalyx, and gram-negative are those group of bacteria which has an extra lipopolysaccharide layer over their glycocalyx. Clear? Clear till this point? Now, further discoveries know that gram-positive bacteria are non-pathogenic in nature mostly. Non-pathogenic means non-disease causing bacteria. Okay. And gram-negative bacteria were more pathogenic. There is a hypothesis regarding this, that why gram-negative bacteria are more pathogenic is that when gram-negative bacteria infect any organism, we have lipid over our membrane. Our cell membrane is majorly form of lipid and protein. Okay. So our immune cell take time to recognize the bacteria which has lipid over their layer. Understood? This is still a hypothesis. This is not yet proved, but this is the hypothesis which is most accepted, that the foreign cells also have lipid layer. We also have lipid layer. So our immune system take time to recognize them. That's why they are easier to cause us disease. Clear? Clear. Now, there is a group of bacteria. Guess. I'm just letting you guess. There is a group of bacteria which is gram-negative in nature and still they don't cause disease. They are non-pathogenic gram-negative group of bacteria.
Yeast?
No, not yeast. Yeast is not a bacteria.
So mycoplasma.
No, not mycoplasma.
Sir, I have to ask.
Mycoplasma.
So the gram-negative bacteria, why are they gram-negative? I didn't understand.
They are gram-negative by nature. Gram-negative was mentioned by Christian Gram that there are some group of bacteria which can be classified based on their
No,
cell
so does that mean
envelope
they're not
structure.
stained? Does that mean they're not stained?
Yeah, based on that stain, they are recognized as gram-positive and gram-negative.
No.
Okay. So the next
Gram-negative
point is
are
that cyanobacteria,
stained or not?
they are not, they are stained, but they lose the stain during alcohol wash
And why do they lose their stain?
because they have lipopolysaccharide layer. We focused on class Bhavya.
No, I don't understand. So what happens? Why does that mean they aren't stained?
Okay, I will, when I will write notes, I will explain it again. Okay. Now, so cyanobacteria is a group of bacteria which is non-pathogenic and they are gram-negative in nature. They have an extra lipopolysaccharide layer over their body. So remember this, you will using this in biology classification. Okay, now write the notes. Now, whatever I am mentioning, just copy the notes. Done, everyone? Done.
Yes.
Now, next point is the classification. So bacteria is classified based on cell envelope. NCERT mentioned it as cell wall structure, cell envelope structure. The first group is gram positive. Another group is gram negative. Now, gram positive have no extra LPS or lipopolysaccharide layer over glycocalyx. On the other hand, have extra thick LPS layer over envelope. Next point, gram-positive are generally non-pathogenic, and these are generally pathogenic. Cyanobacteria is a exception. Lipopolysaccharide, glycocalyx, different layers. Glycocalyx will be present for all bacteria. Lipopolysaccharide will be specifically present for gram-negative bacteria. Glycocalyx will also be present, but LPS layer is not a modification of glycocalyx. Don't mix. Ah,
So gram-negative bacteria are more pathogenic because in our immune system lipids are present, so it is hard to recognize.
yeah, yes, yes. It is tough. This is a hypothesis pronoun. It is not like a proven thing. Okay.
Okay,
They are hard to recognize because of their lipid layer.
okay, sir.
Okay. Okay. One more point. Actually, a question I wanted to ask. That wait, Babel. Wait. One more point that I question wanted to ask, that gram positive and gram negative bacteria, in these two, the gram negative directly obtain nutrition from the host. Okay. And gram positive bacteria will synthesize enzymes, will secrete enzymes, then take up the material or organic substance, then through respiration digest it, then obtain the energy and nutrition. This is what happened now. So in both, out of both of them, which will be having more prominent mesosome as compared to the other one?
Sorry, what's the meanest role?
Which
You said which
Gram
one has more
positive.
prominent what?
mesosome? Which one will have more prominent mesosome? Okay, so gram positive will have more prominent mesosome. Why they will have more prominent mesosome? Because they are not directly taking the nutrition or energy from the host tissue. Understood my point? Understood? Okay, so the third difference is gram positive, more prominent mesosome. It is not
so the
like gram
mesosome-
negative will. It is not like gram negative will lack the mesosome. Okay, gram negative will have the mesosome, but it would be less prominent or less efficient as compared to gram positive.
And so mesosomes are used to absorb nutrients and undergo respiration, right?
They don't absorb nutrients; they just do aerobic respiration. Read the last class notes.
And they're more prominent because they have to gain their food from their surroundings, right?
Yes. Okay. So
As opposed to
we
gram negative, who just
space for one more point. We will cover it later. Okay. Okay. Now the next topic is surface appendages. Now, what are surface appendages? Any structure which arises from the bacterial main body towards outside is considered as surface appendages. Surface appendages consist of three major structures. One is flagella, another one is pili, third one is fimbriae. Okay. Now, out of these three, flagella is the only structure which is involved in motility or movement of bacterial cells. Other two have different function but not motility. Okay, this is the most asked question, one of the most asked questions.
so
Okay.
isn't flagella used for motility?
Yes, flagella is used for motility. Pranav, you were asking.
Used for motility.
Sir, is flagella present in sperm cells also?
In sperm cells.
Yes.
Sperm cells, that is a different, yes, that is flagella, but that is a completely different thing. I am teaching prokaryotic cell, that is a eukaryotic cell.
Yes, sir. I was just asking whether they're the same.
They are not same. Structurally, they are very different, but yes, it is flagella.
Okay, sir.
Okay, so in prokaryotic cell, flagella structure has three different parts. First is basal body, second one is hook, third one is filament. Now, when I talk about basal body, then plasma membrane, cell wall, glycocalyx. Now, in that, the part which is embedded into the envelope, or the part which holds the flagella is called as basal body. The part which connects flagella with the filament is hook. This one is hook, this one is basal body, and this one is filament. Okay, so if I talk about this is the structure, this is the wall, this is basal body which holds the flagella, this one is hook, and this one is filament. Understood? Understood. Now, most of these parts are proteinaceous in nature. And the protein which is majorly involved in the formation of flagella in prokaryotic cell is flagellin. Okay, flagellin is a specialized protein which is present in prokaryotic cells, and they help in the formation of flagella. Clear? Clear. Now, next point. The flagellar structure helps in motility, and they are extension of cell wall. Why they are called as extension of cell wall? Because they directly arises from the cell wall. So it looks like that they are extension of cell wall. Understood this point? Now, in eukaryotic cell, flagella are called to be extension of cell membrane. Why? Because flagella is covered by cell membrane. But in prokaryotic cell, the flagellin protein is helically arranged in a helix manner to form a flagella. Am I
Say
clear?
that again, sir.
Flagellin protein is helically arranged to form flagella. So in eukaryotic cell, the arrangement of flagella is completely different. The eukaryotic cell flagella is completely different from a prokaryotic cell flagella. Clear? Prokaryotic cell flagella is very busy. Next, pili. Pili is another surface appendage which is made up of piline protein. Same, the protein which is associated with pili was named as pili. Okay. Now, what is the major function of piline protein or pili? Pili helps in the formation of conjugation tube. Now, what is conjugation tube? Everyone focus.
Hey, what's the lion?
If this is a bacteria, and this has piline protein or pili, then it can help in the formation of conjugation tube which can attach to another bacteria. If you can think, then bacteria cannot perform sexual reproduction. You know that. If you don't know from now. Okay, bacteria cannot perform sexual reproduction, but still bacteria can perform sexual recombination. Recombination is just exchange of genetic material. Okay, I will teach this in detail in biological classification. But for now, the bacteria which has pili can form a conjugation tube. What is conjugation tube? Conjugation tube is a tubular-like structure which is made from pili to attach to some other bacteria and helps in conjugation. What is conjugation? Conjugation is a type of sexual recombination in bacteria through which bacteria exchange the genetic material. Clear? Clear? Understood? Understood. So this was about pili, but this is the major function of pili. There is one more small function of pili, that pili helps in obtaining nutrition from the host tissue or host cell.
So villi or pili or what?
Pili, P-I-L-L-I. Wait, don't ask in between. I have told you to raise your hand, then ask. Pili. Okay. Now, there is one more function of pili. Pili helps in obtaining nutrition from the other host tissue. Okay. If I say that this is a host tissue and the bacteria is attached to this host tissue, then bacteria can put their pili and from that it can obtain nutrition. Understood? But this is not the major function of pili, but still, pili helps in obtaining the nutrition. So now, go back to gram-positive, gram-negative. In which of them pili would be more prominent?
Gram-negative?
Gram-negative. Okay. So this was the fourth point, that pili is less prominent in pili is less prominent, and in this pili is more prominent. Again, the same thing. It is not like it will be absent in case of gram positive, but it will be less prominent and less efficient as compared to gram negative. Clear? Third structure is fimbriae. You have seen toothbrush. Toothbrush has small bristles. That same structure is fimbriae. Fimbriae are small bristles which are present over the cell wall of bacteria, and this bristle helps in adhesion of the bacterial cell. Whether it is to wall, whether it is to rock, the bacterial cell get adhered to the rock, wall, or the substratum with this fimbriae. Every one of you know the term substratum. Substratum. Substratum is the structure at which or from which a organism attach itself. Like if a algae is attached to rock, then the rock becomes the substratum of that algae. Understood? Understood? Clear? 100%. Okay. Okay. So now you can write surface appendages. The first surface appendages is flagella. Where?
Sir, I wanted to know where the flagellin protein is secreted.
Beta, the whole structure of basal body hook filament is majorly made up of flagellin protein, but the major part of filament is made up of flagellin protein. Other proteins are
what
also
secretes
proteins,
this
not
flagellin
just
protein?
flagellin.
Where does the flagellin protein come from?
It is synthesized by prokaryotic cell.
Okay.
Okay. Okay. If you haven't made the diagram, make the diagram. Make the diagram also. Okay. This small v is basal body. It is a rough diagram, but still make the diagram. Okay, done everyone?
So should we draw the diagram too?
Yes, yes, draw the diagram. It is just a representative diagram. But draw it. Done everyone?
Done, sir.
Okay. Done? Okay. Now, the next topic is bacterial genome. Next topic is bacterial genome. Some part of this I have explained during when I was explaining the prokaryotic cell ka diagram. Now, bacterial genome. Bacterial genome. What is genome? Anyone who can explain the definition of genome?
All of the information coding for proteins and life processes.
No. Anyone who can explain genome? So not total amount of gene, but yet yes, partially
With the
correct.
genetic material of a bacterium.
Genetic material of a bacteria, that's a very generalized statement because genome is term for us also. We can also mention that genome.
So the full complement of the DNA of an organism.
Okay. 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 Okay.
So what is the genetic
Genome is
code?
genetic code. Okay. Listen, genome is haploid DNA content of an organism is genome. From now onwards, remember this statement. Haploid DNA content of an organism is genome. What is haploid DNA content? I am taking human cell example. We have 46 chromosomes. Our haploid chromosomes should be 23. Okay. So 23, the amount of DNA which these 23 chromosome carries is genome. Understood? The amount of DNA which these 23 chromosome carries is genome. Clear? Clear? Clear. Now, we have total how much DNA? Not in length, but in base pairs. You have studied that. We have 6.6 into 10 to the power 9 base pairs. Okay, so this is diploid. If I take haploid, then it is 3.3 into 10 to the power 9 base pair. This is our genome. Understood?
So can you repeat the exact definition?
Genome is haploid DNA content of an organism is genome. Okay, so haploid DNA content is genome. But as we know, haploid diploid is mentioned when there is set of chromosomes. According to set of chromosomes, we mention haploid and diploid. If single set is there, then we mention haploid dual set, diploid 3 sets, triploid, and so on. Okay. But in bacteria, chromosome structure is not formed. The structure which we think of chromosome, like this structure is not formed in bacteria. Why? Why bacteria don't form chromosomal structure?
Because they don't have nuclear membrane.
No, not because of nuclear membrane or nucleus. I have explained that earlier also when I was teaching the structure of bacterial cell. Try to remember those. Doesn't have? No. Not related to reproduction. Why bacteria don't form chromosome? What is a chromosome? Chromosome is a condensed form of DNA. This I can say. Chromosome is a condensed form of DNA. That means the chromosome structure need condensation of DNA to be formed. Who are involved in condensation?
Centrosome.
Bacteria, that's histone proteins, not centrosome. Centrosome are involved in separation of chromosomes.
Oh, sorry.
Histone proteins are involved in, no, not sorry, correct. Histone proteins are involved in condensation of DNA. So histone protein is required if I need most condensed form of DNA. This is the most condensed form possible. DNA cannot. DNA, yes, histone helps in coiling and condensation. Okay, so there can't be any condensation form beyond chromosome. Chromosome is the end. Okay, so if histone protein is not there, there won't be any condensation. But if bacteria has a genome, we cannot say na ki it will have 0 chromosome because it has some amount of DNA. So to explain that or to conclude that, we use a term single chromosome. So if someone asks you, bacteria is haploid or diploid? Bacteria is haploid or diploid?
Haploid?
No, you cannot explain. You cannot explain because no chromosome is formed. But when someone asks you, bacteria have chromosome? Yes, single chromosome. Understood? Understood? Just to conclude things and make things easier, we use a single chromosome term. But always remember, bacteria never forms chromosome. So it cannot be diploid or haploid. Clear? Clear. 1 minute. So this was bacterial genome. Now, bacterial genome has been defined by different terms. Firstly, to make this clear, that bacteria lacks nucleus. If someone asks you, does bacteria have nucleus? Then no. Bacteria lacks nucleus. Nucleus will be always defined when there is a true nucleus. That is, a nucleus with proper nuclear membrane and defined structure. Then only you can use the term nucleus. Other than that, you will never use the term nucleus. Clear? Clear. Now, bacteria lacks nucleus. So what terms we use? The very first and the most common term that is used is nucleoid. Okay. So nucleoid term is used to represent the same structure as a bacterial nucleus. Clear? Second term which is used is genophore. Now, that day also I told 4 means the structure which carries. Okay. So geno term came from genome and genophore term came from the structure which carries the genome. Understood? Understood. Another term which is incipient nucleus means those nucleus or those structure mature. Correct? Correct. Next term which is used is prochromosome. Prochromosome. Now, pro word was used for primitive. So here the chromosomal structure is in its primitive form. So the name term which we give is prochromosome. Clear? Clear? Clear. Now, some of the characteristics. First, it is a double-stranded circular DNA. That day also I explained, if both the ends are joined, then it is circular. If both the ends are separate, then it is linear. Okay, so this is a double-stranded circular DNA. We have linear DNA. We don't have circular DNA. Okay, now, second point, Bacteria will lack the histone protein. That is why the DNA is called as naked. Not because it lacks nuclear membrane. It lacks histone protein. That's why it is called as naked. Clear? Clear. It will have some of the proteins which will be a non-histone protein category. That proteins will help the bacterial cell to properly organize the nucleus. That protein will help the bacterial cell to properly organize the genetic material, but that will not help the bacterial genome to condense to a chromosome. Clear? Clear. Next, bacterial cell will have a high GC content. GC, I guess you know, guanine and cytosine. Okay, so DNA has 4 nucleotides: guanine, cytosine, adenine, and thymine. Adenine, thymine forms double bond. Guanine, cytosine forms triple bond. So which one is stronger? Guanine, cytosine. So the DNA which has high amount of guanine, cytosine, that DNA will be stable more, or the DNA with high amount of A and T?
Guanine and cytosine.
Guanine and cytosine. Okay, so now guanine, cytosine, higher content means the DNA is more stable. So the prokaryotic DNA is more stable as compared to a eukaryotic DNA. Why? Because of the high GC content. Clear? Clear. Now, other than the bacterial genome, first write this. Write till this. Prochromosome and some other names, nucleoid and incipient nucleus. Whenever you are writing double-stranded circular, this is the shorter form. If you are writing it in...
Wait, sir, there's double-stranded here. Just refer to the double helix structure.
Yes.
So like
DNA
if it
will
was single, it would just be RNA, right?
again.
If it was single-stranded, it would just be RNA, right?
RNA can also be present in double-stranded form in some of the viruses.
How?
How means it can exist?
Yes, sir. I had it out.
Continue.
Sir, you said that this bacteria, they form single chromosomes, but how can they even form chromosomes if they don't have histone proteins?
That's what I explained now. Just to make things easier, we mention it as single chromosome because we cannot mention 0 chromosome if there is a genetic material.
Okay.
Understood? So to make things easier that there is a genetic material, we use the word single chromosome.
So it's an assumption.
But 1 minute, I'm explaining something. But the point of chromosome, the proper chromosome is not formed.
Okay. Sir.
Okay, who was asking? Ishan, Pranav.
Sir, the point of having, we assume that it has a single chromosome. It's an assumption. It is not a theory.
Yes. They will never form chromosome. We assume to make things easier. Okay.
Sir,
Yes. Then
what's GC?
GC I explained now. You were not there in the class.
No, I lagged out. My internet's really bad.
GC content is guanine and cytosine content. The DNA which will have higher GC content will be more stable as compared to the DNA which has AT content.
Okay, and histone is protective protein, right?
Not protective, the protein which helps in condensation.
Condensation.
Okay, now next topic. Next topic is, other than this bacterial genome, the bacterial cell have some of the extra chromosomal material. Extra chromosomal material means another DNA or another DNA molecule except this DNA molecule. That DNA molecule is called as plasmid. So the next topic is plasmid. Now, what is plasmid? If I define plasmid, then it would be a double-stranded circular DNA, which is an extra chromosomal material present inside the bacterial cell. Now, why plasmid is important? That's a bigger point. Why plasmid is important? Because plasmid helps in providing some of the unique phenotypic characters to the bacterial cell. Unique phenotypic character means what? If a bacterial cell has some specific kind of plasmid, it will show some specific character. Like a bacteria, let's just take a X bacteria, it is resistant to antibiotic, azithromycin. A X bacteria, if I apply azithromycin, it will not die. Then there is a higher chance that it has a plasmid which is resistant to antibiotic. So these type of plasmid which provide antibiotic resistance are called as R plasmid. Understood? Understood? Understood. Now, second, there are some plasmid which provide fertility to the bacteria. Like there is a type of bacteria which has a plasmid called as F plasmid, and this bacteria has a capability to produce a separate copy of F plasmid and pass it to some another bacteria. This you understood. This bacteria have F plasmid. This bacteria has the capability that it can produce a copy of this F plasmid, and through conjugation it can pass through to a different bacteria which lacks the copy of F plasmid. Understood? Understood. So this is F plasmid, providing fertility to the bacteria. Next type of plasmid is TI plasmid. Capital T, small I, tumor-inducing plasmid. Bacteria cannot induce tumor in animals, but bacteria can induce tumor in plants. Okay, so in plants, by infecting the plant, bacteria can induce tumor. What is tumor? Irregulated growth of cells. Okay, uncontrolled growth of cells. Okay, so this can be induced by some different group of bacteria which have that TI plasmid. Another group of bacteria are called as having RI plasmid. RI means root-inducing plasmid. Some of the bacteria are responsible for inducing root formation in the cell which have just stem cuttings. Understood? Some bacteria can induce the root formation, can synthesize some of the specialized hormones which are responsible for root synthesis. Clear? Clear. So there are different group of bacteria having different type of plasmid, and all those plasmid will be responsible for some or other kind of phenotypic characters. Clear? Clear? Clear. Other than this, plasmid also helps in monitoring the bacterial transformation. Now, what is bacterial transformation? Understand very carefully. This is
responsible for a phenotype?
yes, a different type of plasmid will be responsible for a different type of phenotype. Now, if this is bacteria A and another one is bacteria B, the bacteria A is responsible for a specialized phenotypic character. Let's just guess X phenotypic character. So it has plasmid for that. If I put both the bacterial cell in the same solution, and the bacteria can release a copy of its plasmid into the solution, and the bacteria B can take up the copy of plasmid from the solution. So A becomes the donor, B becomes the recipient. And this genetic material which is shared, this process of sharing of this genetic material is called as transformation. So the B bacteria is transformed after this process, because now it has new set of physical characters, which was there earlier with only A. Now both have those same characters. Understood? So bacterial genetic material, wait a minute, just a minute. Bacterial genetic modification is completely changed. It has new phenotypic character. So if I track down the plasmid, then I can monitor the whole transformation process. Understood this? Clear? Clear. Pranav, you were asking something.
Yes, sir. Sorry to disturb. Maybe I didn't pay attention, but is there a name for the process of exchange of genetic material?
Transformation. It is a type of sexual recombination.
Okay, sir.
Okay. Okay. So I will write the notes now. So this was about plasmid, and this is what plasmid plays a role in a bacterial cell. Okay. And plasmids are restricted to the bacterial cell. They are not found in eukaryotic cell. There is no trace of plasmid in eukaryotic cell. That there are extra chromosomal material, extra chromosomal DNA in eukaryotic cell also, but that DNA is associated with either mitochondria or it is associated with plastids, that is chloroplast. Okay, not like this. It is not freely present. Understood? A cell can have more than one type of plasmid. In bacteria also it is seen the bacteria which is tumor inducing can also have R plasmid. A bacteria which, but if it has different type of plasmid, all the plasmid will fuse to form one structure only. Yes, if a bacteria is there, if just guess this is A bacteria, B bacteria. A bacteria was already having F plasmid. B bacteria passes the R plasmid to this. Then the F plasmid and R plasmid will form a single plasmid. Okay, it will be having fertility as well as resistant to antibiotic. This will be taught in your biotech class in class 12, that how a plasmid structure is formed. This is the complete structure of plasmid. It can have resistant to different antibiotics. A good example of plasmid is PBr322. Okay, this is the plasmid which is commonly found in E. coli. E. coli, you have heard, Escherichia coli. Most common example of bacteria. Okay, so in that PBr322 is found. That PBr322 is used for different biotech processes, biotech applications. That is an example of plasmid. Like that, number of plasmid examples are there. You will be taught the whole plasmid structure in the class 12 part. Clear? Clear? Clear. So this was about plasmid. Copy. You asked me something.
So what did you write in the second one?
Double-stranded circular naked DNA.
So naked refers to the fact that it does not have histone protein, right?
Yes, yes, yes. Wherever you see naked, it is related to histone protein only. You will never see a naked with respect to a eukaryotic cell.
So the TI plasmid for tumor inducing, right?
Yes. Done. Done. Done, everyone? Everyone done?
Yes, sir.
Okay. Next topic is ribosomes and inclusion bodies. Now, this is the last part of prokaryotic cell. Ribosomes and inclusion bodies. Now, ribosomes are also called as protein factory of the cell. Okay, ribosomes are also called as protein factory of the cell because they are the sole structure which is responsible for protein synthesis. No other structure inside the cell can synthesize protein except ribosomes. Even if some other mitochondria, chloroplast, endoplasmic reticulum, whoever is synthesizing protein, it is directly or indirectly associated with ribosomes. Okay. Now, ribosomes are the protein factory, first point. Second point, ribosome are non-membrane bound structure. Okay, always remember ribosome will not be bounded by any membrane. It will not be covered by any membrane. It can attach to a membrane, but can never be covered by any membrane. Clear? Clear. Third point, ribosomes are made up of rRNA and protein. I think you have heard about 3 types of RNA. If you haven't, you will study that in 12, but I will give you an overview. There are three types of RNA. rRNA or ribosomal RNA. These RNA helps in structural part, formation of the structural part of ribosome. Next is tRNA or transfer RNA, which helps in translation, that is the formation of protein. Okay. Third one is mRNA or messenger RNA. This RNA helps in passing the genetic information from DNA to protein. Understood? You have heard about transcription and translation. Transcription is formation of RNA from DNA and translation is formation of protein from RNA. Understood? So from the RNA, from that RNA from which protein is formed, that RNA is called as mRNA. Clear? 100%? Clear? Clear. Now, moving on. So ribosomes are made up of rRNA and proteins. There can be a different kinds of 80 to 82 types of proteins are involved in ribosomal formation. That more about protein we will study in the eukaryotic ribosome. We will mostly cover the prokaryotic part of ribosome now. Okay. In prokaryote, there is a type of ribosome which is 70S type of ribosome. Okay. 70S type of ribosome has 2 subunits, 50S and 30S. Now here math is not matching. 50
Hey,
plus
but the
30
math doesn't count.
50 plus 30 equals to 70. Okay. Now, how we get that S? S is Swedberg's constant or sedimentation coefficient. Okay. Now, what is that S? Everyone listen carefully. If I take a test tube with gradient, okay, I am forming a gradient. I can take sucrose ka gradient or cesium chloride ka gradient. Okay, so if I take a normal gradient here, 100%, 90, 80, 70, 60, 50, 40, 30. Like this, gradient will increase. Okay, now I put a cell culture here, the extracted part of cell without the cell wall and plasma membrane. Understood? Understood. Now, I put this test tube under a centrifuge. And now that centrifuge will be called as differential centrifuge. Why? Because I am taking a differential gradient. Understood? Understood? Understood. Now, it is also called as gradient centrifugation. Clear? Now, if I rotate that in centrifuge, the part of this cell culture will get separated according to their density. This you understood? The part of the culture will get separated according to their density. That means if some structure has a density of 70, then it will be separated here. Some has a density of 50 or 30, then it will be separated like this. Understood? Understood? Now, so when Swedberg, the scientist, used the cell culture and took ribosomes with both subunits together, then that subunit was separating at 70. So he called that subunit as 70S. And when he separated two of the subunits of 70S, break the subunits, remove the part and in two different parts, and then he again put the cell culture, then he found one subunit at 50 and another at 30. Understood? Understood. From this, he deciphered that there are two subunits of ribosome. One is larger dome-shaped, a cap-shaped subunit, and another one is smaller dome-shaped subunit, just like this and this. Okay, so when he used both subunits together, it was dense. It was showing density around 70. Okay, for prokaryotes. When he separated both the subunits and again did the centrifuge thing, then he found out that one subunit is a larger subunit, which is there at 50s, and one subunit is a smaller subunit, which is there at 30s. Clear? Clear? Clear. So he separated both the subunits and found out these density numbers. Clear? Clear. So this was density centrifugation, through which we know that the ribosome is individually 70s, and when I separate both the subunit, then larger subunit is 50s and smaller subunit is 30s. Okay, I will explain this again in the eukaryotic ribosome. Just remember it for now. Okay, next, important part of ribosome. If we talk about prokaryotic ribosome, then prokaryotic ribosome can be showing two different associations. This is plasma membrane of prokaryote. This is cytoplasm of prokaryote. The ribosome, when they are not synthesizing protein, when the ribosomes are not synthesizing, not doing anything, then both the subunits are freely present in the cytoplasm. Understood? But when ribosome will synthesize protein, then both subunits will come together, attach themselves, and then start to synthesize protein. This is clear? Clear. Now, when ribosome wants to synthesize a secretory protein, the protein which is needed to be secreted out of the cell, then ribosome associate themselves temporarily to the plasma membrane. And if the ribosome is synthesizing some protein which is not destined to secrete it out, then it will be called as non-secretory protein, and those ribosomes will be present in cytoplasm. Understood this? So ribosome can be
In the
present.
case of secretory proteins, where are they present?
Wait, I am repeating it again. Ribosome can be present in two places. If ribosomes is associated with plasma membrane, the association is temporary, and that association will be for the secretion of the secretory protein, will be for the synthesis of secretory proteins. Okay, and if the ribosomes are present in cytoplasm, then they will be responsible for the synthesis of non-secretory proteins. Clear? But wherever the ribosome is present, the two subunits will only be together when there is protein synthesis. If there is no protein synthesis, then there is no separation between the two subunits. Clear? Sorry, no attachment with the two subunits. Clear? Clear? Clear. This was about ribosomes. One last point about ribosomes, that we already know that from mRNA, proteins are synthesized, and on this mRNA, ribosomes are associated, which are responsible for the protein synthesis. Now, in prokaryotic cell, at the same time, more than one ribosome can be associated with single mRNA strand. What it helps? This part will synthesize protein, this part will synthesize protein, this part will synthesize protein. Or this ribosome can synthesize protein till this can synthesize protein till this. Understood? So from the same mRNA strand, I can get more than one protein at the same time. Higher efficiency. Higher efficiency. But there is a higher chance of error also. So this is the primitive character, but it helps in higher efficiency for translation in prokaryotes. And this structure, when one mRNA is associated with more than one ribosome at the same time to synthesize more than one protein, this structure is called as polyribosome or polysome. Polyribosome or polysome. Polysome. Okay. Understood? 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 Okay. Now, you know the word some. Some means, some or soma means body. Where does the ribo word came from? Ribosome. Some means body. Ribo means? Ribo term came from?
Latin?
No. What did you told? Bhavya, what did you told?
The ribo term come from Latin by any chance?
No. Ribo term came from, I have just told you. I have explained during the explanation only.
Oh, wait.
RNA. RNA. Okay. Ribonucleic acid, RNA. Ribo term came from RNA. Okay. Start writing. Ribosomes.
So what are inclusion bodies?
I haven't taught yet, beta. I will teach. You can make diagram also if you want. Large, small. Huh? Cytosol is another name for cytoplasm. Cyto means cell, sol means solution. Okay. Huh? Who asked?
What is the word before in cytosol?
Present.
Oh,
Those present in cytosol synthesize non-secretory protein. Don't use, those who are joined online also, don't use these shortcuts in your theory exam. These shortcuts should be restricted to your note copy. Okay.
Which
I am
does-
using
does
this
any
because this is
classes have
easier
to
for
be-
me. Done? This one done? One more point is there. Done? This is done? Now, one more point. If one mRNA strand. Aware. Continue.
Sir, what's the difference between rRNA and mRNA?
rRNA is a structural molecule. It will be part of ribosome ka structure. And mRNA will be the RNA molecule which is formed from DNA to form protein. Done? Done. Now,
So one more.
next topic is inclusion body. What is inclusion body? Inclusion means to include. Okay, so those all pre-reserved material which is included inside the cytoplasm are inclusion body. As simple as that. Okay, so all the reserved material which is freely present in the cytosol or the cytoplasm are considered as inclusion body. It can carry phosphate granules. It can be synophysian. Synophysian means starch granules. Okay, it can be synophysian granules. It can be lipid globules. It can be glycogen globules, or it can be other inclusion bodies also. Okay, but remember, these inclusion body will also be non-membrane bound. There won't be any membrane over these inclusion bodies. Next, a different kind of
What inclusion bodies? Say what inclusion bodies?
all the pre-reserved food material which is present inside the cytoplasm. Okay. Now, next, there is a different kind of structure which is present inside the cytoplasm, but are considered under inclusion body are gas vacuoles. The bacteria can intake gas and can form gas vacuoles. But vacuoles, when we will study the same vacuole in eukaryotic cell, then it will be having a membrane over it. But if I am studying vacuole in a prokaryotic cell, then again it will be a non-membrane bound structure. Understood? So there can be gas vacuoles like carbon dioxide gas, sulfur gas can enter the cytoplasm and can form gas vacuoles. These gas vacuoles are nothing but inclusion body, helps the bacterial cell to provide buoyancy. Understood? Buoyancy to those bacteria which are aquatic in nature and which floats on the water surface. Clear? Like cyanobacteria are there.
Say that again.
Purple green sulfur bacteria are there. Okay, so these are different groups of bacteria which have gas vacuoles, and gas vacuoles provide buoyancy to them. Okay, okay, right.
So what's a gas vacuole?
Gas vacuole is an inclusion body.
So the ones in the blue are examples of inclusion bodies, right?
Yes. These are different types of inclusion bodies which are present in the bacterial cell.
So the second one, is it cyanophysian?
Yes, cyanophysian granules. It is actually a starch granule. But as it is found in cyanophysi group, so it is called as cyanophysian granules. Done?
Okay. Sir,
I
just wanted to clarify what cytosol is.
Cytosol means cytoplasm. Cyto means cell. Solution which is present inside the cell is cytoplasm. Another name for cytoplasm.
Okay, alternative name for cytoplasm.
Alternative name. Cyto means cell. Plasma. Plasma means plasma. So cytoplasm. Cytosol, solution. Cyto, cell.
Okay, sir, thank you.
Done. Done. Now, those cells which have true nucleus are called as eukaryotic cells. EU means karyotic nucleus. Okay. Now, eukaryotic cell will be an advanced group of cell which will be having membrane-bound structures or membrane-bound organelles. Because of the presence of membrane-bound organelles, eukaryotic cell shows extensive compartmentalization. What is compartmentalization? If I draw a eukaryotic cell, each of the membrane, each of the organelles will have its own membrane. This is one compartment, this is another compartment, this is another compartment, this is another, this is another. So number of compartments are formed due to the formation of membrane-bound organelles. So in eukaryotic cell, we will see extensive compartmentalization. We will see extensive compartmentalization in eukaryotic cell. Now, next point. Unlike in prokaryotic cell, the fundamental structure was similar for all the prokaryotic cells. But in eukaryotic cell, the fundamental structure is different. Different prokaryotic cell can have different organelles, can have different basic structure, can have different types of membrane, can have different cell organelles. So the basic fundamental structure of all eukaryotic cell is not similar. Okay. Now, prokaryotic cell was much smaller in size, eukaryotic cell are much larger in size. Like eukaryotic cell, the average 10 to 20 microns. Okay. Next, eukaryotic cell includes the other four kingdoms except the molecular kingdom. Eukaryotic cell is Vista, fungi, plants, and animals. Now, in eukaryotic cell, I will start with cell wall. NCERT starts with cell membrane, but I will start with cell wall. Okay. So the first structure for eukaryotic cell is cell wall. Now, cell wall is a living structure or a non-living structure? Non-living structure. So we define cell wall as rigid non-living layer which is present over the plasma medium. And this layer, that is cell wall, is not universal in nature. They are present in some of the groups in case of eukaryotic cell. Major groups are fungi and plants. Other than fungi and plants, some of the protista also have cell wall, but we won't consider them because majority of protista lacks the cell wall. Clear? Now, cell wall based on the developmental time, like where the cell wall is formed during the early development or cell wall is deposited during the later development stages, cell wall is classified into two types: the primary cell wall and the secondary cell. Okay. The composition which I will tell are of primary cell wall. If I mention about fungi, then fungi have cell wall made up of chitin. Okay, and chitin is a homopolymer of NAG. What is NAG? N-acetyl glucosamine, which was there for peptidoglycan. But that time it was acting as a heteropolymer as a part of that peptidoglycan. But here only NAG forms the chitin. Where we have seen chitin other than this fungi?
Cell wall of fungi.
Other than fungi, where we have seen chitin?
Cell wall of fungi.
Other than fungi, where we have seen chitin?
Proctists.
Where?
Proctists.
No. Any example where we have studied about chitin?
Sir, can you repeat what you want? I'm not able to hear properly.
I was saying that chitin is present as a cell wall composition for fungi. Other than fungi, where chitin is formed very commonly formed, which we study. You must have heard somewhere about it. Chitin somewhere, part of some skeleton.
In mollusk,
It is part of some skeleton. It is part of exoskeleton of insects.
arthropods. Arthropods.
Yes, arthropods. Correct. Now you are right. Okay. So exoskeleton of arthropods or exoskeleton of insects. Chitin is a major component of that. Okay. Okay. I will give you a very bad example. Chitin is a very tough structure. That's why when you kill a cockroach, it sounds like a crisp because of that chitin. Okay. So chitin is a very tough structure and very fragile structure. Okay. But this cannot be seen in fungi because fungi are very small insects. So chitin is a very important component for fungi as well as the arthropods type. Okay.
You mentioned a fungi example. What is that?
Fungi example.
I mean the homopolymer of energy. What is that?
Chitin is a homopolymer of energy.
Oh.
Another.
The same one in arthropods.
Another.
Yes, sir. I was going to ask what a homopolymer is.
Okay. Homopolymer is there is a single monomeric unit.
Oh, okay.
If
Yes,
there
sir.
is more than one type of monomeric unit, then it is heteropolymer, like peptidoglycan.
Okay. Thank you, sir.
Pranav, you were saying something.
No, sir. I was only saying that chitin is the same one which is there in fungi and arthropods, which you mentioned before.
Yes. Chitin is the same. Other proteins or other things can be present, but chitin will be the same. One more thing for everyone is galactanes and mannans. You have heard about galactose?
Yes, sir.
Galactose, sugar galactose. Okay.
Sugar.
If that sugar is modified into an alcoholic sugar, then it will become galactanes. Understood? And mannose. Mannose is another form of galactose sugar, and that mannose sugar is alcoholic functional group. It will form mannose. So both galactanes and mannans are alcoholic sugars.
Sir, how do you spell galactans?
Galactans.
Okay, sir. Thank you, sir.
Another, if you still have doubt.
Oh no, sir, I forgot to lower my.
So these two, because in fungi there is no classification of primary wall or secondary wall, but in plant there is a classification. So these are the composition of primary cell.
Sir, what is the one after cellulose in higher plants?
What is the
word
cellulose?
after
Heavy cellulose.
hemicellulose?
Yes, modified form. So these are some of the composition which you need to remember. Okay. After this, we will start with what are primary cell wall and what are secondary cell wall. Done, everyone?
Yes.
Done. Now, next topic is primary cell wall and secondary cell. Primary cell wall and secondary cell wall is classified based on the developmental stage when it is formed. If a plant cell is newly formed, then it will start to synthesize primary cell wall during its early development. Okay. It will start to synthesize primary cell wall during its early development. Okay. This is a plant cell. It will synthesize the primary cell wall. Now, the primary cell wall will grow with the plants. What that means? If this is a plant cell and it has primary cell wall over it, then it's obvious that plant cell wall will increase the cytoplasm during the development. So if it will increase the cytoplasm, then the plant cell wall should break, but it doesn't break. Instead, it grows and it extends with the extension of the plant cell. Understood? So the primary cell wall is capable of growth and it is extensible in nature. It can extend with the growth of the plant cell. Understood? Understood. Second point, plant cell. Plant cell, sorry, not plant cell, primary cell wall. Primary cell wall shows growth and it is extensible in nature. Plus, primary cell wall is permeable in nature. It allows most of the solutes and water to pass. It is not impermeable in nature. Okay. The structure which is semi-permeable or selectively permeable is glass wall. Cell wall doesn't select any solute. Cell wall will allow all the solutes which can pass through it. Because it pass through it means the size. Okay. It will not allow some larger macromolecules, but it will allow most of the solutes, whether it is ions or something. Clear? Now, next point, Pranava.
Sir, you said about the cell wall growing with the cell and not tearing. So that is called a delimiting structure, right?
Yes, that's why it is called as a delimiting structure. Okay. Now, next, secondary cell. A cell wall which is deposited during the later life stages of a plant cell. When a plant cell is going to mature, like a plant cell is going to perform a specific functions. During that time, it will differentiate and mature itself. During that time, secondary wall is deposited. Where secondary wall is deposited? Secondary wall will be deposited inner to the primary. Why? Because it's obvious that plant cell.
Wait, what? Secondary what is deposited, sir?
Secondary wall will be deposited towards the inner side of the primary wall. Secondary wall will not be deposited outside because it's obvious that plant cell will not secrete across the primary cell wall and then deposit. Okay. Now, but remember that primary cell wall eventually get degraded or it diminished during the maturation. And during after completion of maturation, when the cell is completely mature, then the primary cell wall will be replaced by the secondary cell. Understood? But secondary cell wall has some negative aspects. Like now the cell is dead because of secondary cell wall. Why? Because secondary cell wall is impermeable to water. It is impermeable to all the solutes and water. So if a cell is getting matured, like xylem tracheids or xylem vessel cells, and it is depositing the secondary wall, then eventually it will become a dead cell. Understood? Another example is sclerenchyma cells. Sclerenchyma also, when it is young, it will have a thin wall. But eventually, when it matures, it will start to deposit layers of secondary wall. And when the secondary wall deposition is completed, then it will become dead and there won't be any exchange of material or living protoplasm inside. Where mute yourself. Okay, so there won't be any structure which will be present living inside the cell. Clear? Am I clear? 100%? No. Secondary cell wall is formed like primary cell wall was majorly deposition of cellulose, hemicellulose, galactose, calcium carbonate, and protein. But secondary cell wall is formed of two depositions. The first deposition is that suberin. Second deposition is lignin. If suberin is deposited, it is called as suberized. If lignin is deposited, it is called as lignified. Understood? If a cell has suberin deposition, it is superized. If a cell has lignin deposition, it is lignified. Which cell shows which deposition? This we will cover in the anatomy of the plant.
And sir, most living cells only have the primary cell wall, right?
Yes. Okay. So this was the explanation for secondary cell wall and primary. Understood? Understood. Any example of cells which will have primary cell wall? Meristematic cells, any other cell which is not dividing still, which are permanent cells, still they will have primary cells. Any group of cell which is not dividing or permanent cells, still they have.
So root है.
Lenticels.
Root.
Root cells, no, group of cells. Root is a part, no?
Lenticels.
Someone was saying something. Collenchyma, okay. Parenchyma, correct. Okay. Parenchyma, collenchyma, both are good. Collenchyma will have a thick deposition, but still it will be a living cell. Okay. Now start writing. 1 degree is primary. This is
Sir?
primary. Yes.
What do you mean by degenerates during maturation?
When the secondary cell wall is deposited, then primary cell wall eventually degenerates.
Okay.
It is like incomplete cell, in completely matured cell, you won't find any primary cell.
So when the secondary cell wall forms, the primary cell wall degenerates.
Yes, it starts to degenerate.
So but what about the cells without the secondary cell walls?
What are the?
What about the cells without the secondary cell walls?
They are not completely matured yet. Like parenchymatous cell. Parenchymatous cells still have the ability to de-differentiate and form new cells.
Okay. So, but then if they fully mature, then they get the secondary cell wall and then they die.
So dead cell also can show function. The xylem, most of the xylem materials are dead.
Okay, okay.
It is not lignified, it is lignified. I missed the G. Now, next structure which is associated with cell wall is done, everyone?
Yes, sir.
Okay. Now, middle lamella. Lamella means layer, middle means in between. So the layer which will be present between two adjacent cells will be called as middle lamella. As simple as that. Okay. Now, where middle lamella will be present? If I draw, this is one cell, this is another cell, this is just a secondary wall, here to this primary wall is present. The deposition which will be present between the adjacent cells will be called as middle. Sometimes middle lamella is also called as tertiary wall. Okay. but it is mentioned with respect to that it is an extra layer over the secondary. Understood? So middle lamella holds the adjacent cells. It is just as of cement for the bricks. To hold the brick, we put cement in between. Okay, to hold the bricks in the position. Same plant cell uses middle lamella to put between the cells and to hold the cell. Okay, now middle lamella is made up of, or when I define middle lamella, I define it as a amorphous cementing material. Amorphous means the material which don't have its own morphology. Understood? Amorphous. Same as of cement. Wherever I will put, it will take the morphology of that structure. Okay. Next, cementing material. It helps to cement between the cells, so it becomes cementing material. Clear? Next point. Middle lamella as a structure is made up of calcium and magnesium pectate deposition. Okay, middle lamella is a deposition of calcium and magnesium pectate secreted by the plant cells. Clear? Now, middle lamella eventually degenerates during the ripening process. That's why if you take a raw mango, it is very tough. You cannot put finger. You cannot press it and it does not have a soft texture. But if you take a ripened mango, it has a very soft texture. It has a very gummy texture. Why? Because earlier when it was raw, that time middle lamella was properly developed. Understood? So all the cells were there in their position. But when it ripens up, the middle lamella get dissolved. So now when you put finger, it feels like soft. Why? Because the cells are getting separated. The more pressure you will apply, more cells will separate. Understood? Understood? So middle lamella is a structure which provides rigidity to the plant. Okay, and it holds the cells in place. Which structure or which chemical does the same thing in humans? Which structure does the same thing as of middle lamella in humans? Think.
Sir,
Yes.
the lamina is in the innermost part of the cell membrane of the cell wall, right?
No.
Then
Lamella is a general term used for layer.
oh, so the inner lamella is the innermost layer of the cell wall.
If you are defining the cell wall, why you are using the term inner lamella?
So deposition of calcium and magnesium, what?
Calcium and magnesium pectate.
So what is calcium magnesium pectate?
It is a chemical which is secreted by plant cells to form the middle lamella. Just as of
Okay.
collagen does the same thing which middle lamella does in plant cell. Collagen eventually starts to degenerate when we age. That's why we have wrinkles. Wrinkles is a result of absence of collagen because there is no one to hold the cell. So tissues form wrinkles. Understood?
Sir,
Yes.
so the lamella which contains collagen degenerates.
Middle lamella.
No, I mean the collagen. Collagen, I mean.
Collagen is there in animals, not in plants.
Okay, so only in animals, when the collagen degenerates, wrinkles start appearing.
Yes. Collagen is the most abundant animal protein in the world. Okay. If you haven't heard it, hear it. Collagen is the most abundant animal protein in the world. Okay. So you will study this in biology. It is there in your science. Okay, so this was about middle lamella. Now, an important point about middle lamella, that if middle lamella and cell wall are present, then there should be something which should form a connection between the cells, so that cell can easily share the nutrient, it can share the water, or it can share the ion through that connection. So that living connection is formed by a structure called as plasmodesmata. Okay, what is plasmodesmata? Plasmodesmata is a transverse living symplastic connection between the adjacent cell, and it is a cytoplasmic connection. Cytoplasmic connection means it is such huge in size that cytoplasm can be shared between the two cells. Understood this? Understood this? Now I will make a diagram for you. Okay. If this is secondary wall, inner to that we have primary wall. So primary wall is thick, that means cell is not completely mature. Inner to that we have primary wall. Between that we have middle lamella, and when there is a transverse between the cell wall and middle lamella, this cytoplasmic connection between cells is called as plasmodesmata. Understood this? Understood this? No. This will be bordered by plasma membrane. The bigger question is that how plant cell forms this plasmodesmata. So I am making it here. If this is a plant cell, it has divided its nucleus and now it is going for cytokinesis. So when it is going for cytokinesis, there is still an endoplasmic reticulum present in between, and that endoplasmic reticulum is not separated properly between the two cells. So it is present in between. So plant cell will not break the endoplasmic reticulum. It will form the cell wall surrounding that endoplasmic reticulum. Understood? So now when the cell formation is completed, then here cell wall is formed and endoplasmic reticulum is present in between. This structure which is formed will form the plasma. Understood? Understood everyone? So this structure which is formed here will form the plasma. So endoplasmic reticulum plays an important role in the origin of plasmodesmata. Understood? And plasmodesmata is called as symplastic living connection. Why symplastic? If these are cells of plant is absorbing water from here and it needs to pass it to the zygote, then plant have two options. Either plant will pass the water from the space between the cell wall and plasma membrane. Okay, that means cell wall is assisting for the transfer. Then it becomes a dead path. Why? Cell wall is dead. Understood? But plant have a second option to pass the water through the cytoplasm. Then it will become a symplastic or living path. Understood? Because it is passed across the living component of the plants. Clear? This is clear. That's why plasmodesmata is called as symplastic living pathway from which plant shares the cytoplasm, or it is a connection between the adjacent cell for the movement of nutrition and other molecules. Clear? This was plasmodesmata. Now, next point. There are some structure other than plasmodesmata which is formed during the maturation process. Eventually, they will degenerate, but they are formed during the maturation process for the continuation of the metabolic process. Like, if I draw these two layers as primary wall, these are two adjacent cells. This is the cell wall, this is the cell. Now, during the maturation process, they will deposit the secondary wall. And eventually, this secondary wall, the thickness will increase. Okay, but during the process, there are some parts of secondary wall which is not properly deposited. That means the formation of secondary wall is not continuous, and there are some space in which secondary wall is not deposited. From there, both the plant can exchange some of the solutes and water. Understood? When it is still in living condition. So these places at which secondary wall deposition is absent are called as pit, P-I-T, pits. Understood? It is very different from the plasmodesmata, because plasmodesmata is a complete living connection to which two plant cells shares the cytoplasm. And through it, plant cells are restricted to share some of the solutes and water molecule. Those which are allowed from the primary wall to pass. Clear? Any doubt? No. Okay, start writing. Don't draw this. First write the notes, then draw this. Okay, now draw the structure. The tubule is this thin tubular part which is formed by endoplasmic reticulum.
Sir, this tube, what's it used for?
It is used for exchange of cytoplasm
Oh,
and
so
other
you're saying,
nutrients.
so you're saying all plant cells basically share one cytoplasm because they're going through them?
Not all plant cells, but yes, if they are living and they have some of the connection, then it must be plasmodesmata.
And they're used for communication too, right, sir?
Communication, but transfer of nutrients and water.
Wait, sir, what are you saying? Communication?
Not used for the communication. No? It is used for transport of nutrients and water.
The transport of what?
Nutrients and water. Then,
Sir, can you give me one more?
whenever you are drawing a diagram, use pencil. Okay. Always use pencil. Don't use pen for diagrams, at least for biology. This is chemistry, but for biology, always use pencil.
So once CW and 2CW refers to first cell wall and secondary.
Primary. 1 degree is primary, 2 degree is secondary.
Okay. Primary and secondary cell.
Yes. Cell wall. Okay, done. Now, after plasmodesmata, right click.
Sir, can you go back just 1 minute? Done, sir.
What's deposition, sir? What's a deposition?
Deposition is absent. Yeah, deposition is absent.
No, deposition of what, sir?
Secondary cytoplasmic position. So it will be lignin or superin.
Sir, can you speak a bit clearly? I can't understand what you're saying. Sir?
Deposition of lignin or superin. Deposition of lignin or superin. It is secondary cell wall deposition, so it must be either lignin or it will be superin.
Secondary cell wall deposition is not continuous.
It will be absent somewhere.
And will be absent where the plasmodesmata is, right?
No, plasmodesmata is not related to pits. Pits are different. Plasmodesmata are different.
Pits or pills?
Pits. It is PITS. Pits. Done. So this was about cell wall. Next topic is cell membrane.
Sir,
Yes.
is there a function of pits?
It is like that. It acts as a way of connection between two cells. When the cells are not completely mature during the maturation process, eventually it will diminish if it is a completely matured cell. Okay. Or even if pit is present, then for a dead cell, pit will act as a point of exchange for water.
Okay, sir. Thank you, sir.
Like if you will study sclerenchyma, then sclerenchymatous cells have pits. That pits act as an exchange for water. Now, cell membrane, next structure of eukaryotic cell. Cell membrane is a structure which is universally present. I have already told that. Okay. This structure, the fundamental structure of cell membrane will be same for all the cells throughout. There won't be any exception, but composition can differ. The composition of cell membrane can differ Generally, the cell membrane has three components: lipid, proteins, and carbohydrates. Other than these three, some other chemical can be present, but majorly these three will be present. The composition difference is in the percentage of lipids and proteins. Like if I take an example of RBC membrane, red blood cells membrane, okay, then that membrane has 52% protein and 40% lipid. And
So,
other, yes,
to confirm, pits are just parts of the cell wall which are like thinner, right?
but I will explain it later on. Let me complete first. Okay, cell membrane. Cell membrane, the red blood cell or the erythrocyte cell membrane has 52% protein and 40% lipids. Okay, now other 8% is composed by different carbohydrates are there, different proteins are there. Okay, but I am saying that whenever cell has more requirement of proteins, then it will have a higher percentage of protein as compared to lipid. And if the cell requires, if the component require more amount of lipid, then it will have more percentage of lipid components as compared to protein components. So the composition can differ, but the basic component of a membrane will remain same, that is lipid, protein, and carbohydrate. Clear? Clear. This was started earlier. Many model came. Robertson unit membrane model was the most famous model. After that, when there was advent of microscope, advent means discovery of microscope, during 1950s, we saw that the structure of plasma membrane has two layers of lipid and proteins are present in middle. Okay, this study was done when electron microscope was discovered. After that, number of scientists came and they explained about the composition of plasma membrane. And most of the study about chemical composition of the membrane was done on RBC or erythrocyte. Why RBC or erythrocyte? Why they become the best material for the composition of plasma, to study the composition of plasma. Why? It was a very easy answer. Very easy answer. You have studied about, not this, but you have studied about RBC. You have studied about RBC. Think. RBC lacks membrane-bound organelles. RBC lack nucleus. They don't have mitochondria. They don't have lysosomes. They don't have endoplasmic reticulum. Why? To accommodate more and more hemoglobin, so that it can more and more oxygen. It can transfer more and more oxygen. Okay. Now, if I take a cell with number of organelles having plasma membrane, crush it and use that sample to study the composition of plasma membrane. And on the other hand, I take a RBC, crush it and use that sample for studying the plasma, which will be less contaminated. RBCs will be less contaminated with other membranes. Okay, that's why RBCs are used, because they don't have any excessive contamination of the other organs. Understood? Understood. Now, next point. When we deciphered, we saw that lipids are the major components. After that, protein, then carbohydrate is present. And in lipid, there were number of forms of lipid, like phospholipid was there, glycolipid was there, sphingolipids were there, cholesterol was there, and other forms of lipid was there. But the major was phospholipid. Phospholipid forms the major part of the lipid component of the plasma. Understood? Understood? Next was protein, then it was carbohydrate. Using this all knowledge, in 1972, two scientists came forward, Singer and Nichols. Okay. Both of these scientists proposed a model called as fluid mosaic model. Fluid mosaic model was proposed to explain the structure of membrane. Before Singer and Nichols, total 8 models, but none of the model explained the dynamic nature of the membrane. Dynamic nature means the membrane can change itself according to the requirement. It has a flowing nature. It has a dynamic nature. Understood this? Understood this? Now, Singer and Nicholson explained that dynamic nature of the membrane through fluid mosaic. So their model became the most accepted worldwide model for plasma membrane, and till today there is no exception to that method. Still we study the fluid mosaic model of Singer and Nicholson. Recently in 2023, Nicholson died. Singer died earlier. Recently in 2023, Singer died. Nicholson died. Okay, so they were the most famous scientists to explain the membrane structure. Understood? Understood? Understood. Any scientist which you have studied who recently died in 23 or 24, or I think 25, July 25. Very famous scientist. You must have heard his name. Very famous scientist associated with DNA. Associated with DNA. Crick died. Okay, Crick died recently in 2021. Okay, so cell membrane, right?
So, so RBC is used for composition study of cytoplasm, right?
Here I have mentioned cytoplasm.
No, then what? Oh, you meant RBC is used for composition study of cell membrane?
Yes, yes. Was asking something.
Yes, sir. What's after glycolipids?
What are glycolipids?
And what's after glycolipids?
Sphygmolipids. SPHIGNO. Sphygmolipids. When choline molecule is attached with phospholipid, it forms a sphygmolipids. It will be there in your biomolecules chapter. The whole structure of all these four types of lipid will be there in your biomolecules chapter. Okay. Done, everyone? Done? Yeah. Now, next point was these all years are given in NCERT, so you need to remember these all. Yes? Done. Now, I missed one point in cell wall. Functions of cell wall. Okay. So after I will complete this part, then I will explain that. I have explained that in prokaryotic cell last class. Okay. But two more points are there mentioned in eukaryotic cell wall that I will mention. Okay. Moving on with the plasma membrane. Now, when I talk about plasma membrane has major component as lipids. Okay. So if I draw a structure of a lipid molecule, I draw a structure of a phospholipid molecule, then it has a tadpole structure. Tadpole structure means it will have a head and it will have a tail. Okay. Now, this tadpole structure will have a phospholipid head and a saturated carbon chain. I guess you understood the term saturated.
So what are you referring to? What are you talking about, sir?
Lipid.
A lipid. The structure
Yes.
of a lipid. Okay.
Single bonded carbon molecules, when it forms chain, it forms saturated hydrocarbon chain. Most of these chains are fatty acid chains. Okay, fatty acid means when a straight carbon chain is attached with a COOH molecule, that is carboxylic acid, it forms a fatty acid. Okay, so most of these are fatty acid chains. Now, the head part, which is made up of phospholipid, is actually polar in nature. Polar means it will be hydrophilic in nature. Water being polar molecule, polar attract, non-polar attract. Non-polar repel each other. Okay. Now, this phospholipid molecule is directly bonded with two of the fatty acid chains. But the point is that phospholipid by nature is polar and hydrophilic in nature. On the other hand, when I talk about the tail, then it is hydrophobic or non-polar in nature. That means the tail cannot get associated with water molecule. Understood? Understood? Understood. Now, taking an example, this part is the head, this part is the tail. If the membrane is present like this, then this tail part will interact with the water molecule present in the cytoplasm. If membrane is like this, then the tail part will interact with the outside aqueous environment. So membrane should be somewhere like this. That's why plasma membrane has a lipid bilayer. Okay, so one molecule of lipid and another molecule of lipid is present like this. Now, both of these sides have head. They can interact with water, and tails are projected towards the inside. So that tail cannot interact with water to avoid the interaction between the tail and water. Understood? Understood. So that's why lipid molecules are arranged as a bilayer in the membrane to avoid the interaction between the tail and the water molecule. Clear? Clear. Now, lipid molecules are fluid in nature. By fluid, I mean not like water, but like jelly. Okay, jelly-like. The term used for lipid molecule by Singer and Nicholson was quasi-fluid nature. Quasi-fluid means fluid-like nature. It will not be like if I put it on table, it will flow. But it will be like if I put it on table, it will remain as it is. But if I require it, that it should show a dynamic nature, that it should flow or it should move according to my requirement, then it can do so. Understood? Understood this? That means our membrane, which we have inside the cell or wherever it is present, that shows a dynamic nature. That lipid molecule can move themselves according to the requirement. Understood? Understood? This is fluidity. So if I define fluidity, fluidity means movement of molecule within the cell membrane is fluid. Clear? Clear? Clear. By this fluidity, lipid, remember lipid provides this fluidity. Lipids is the only molecule which is responsible for fluidity. Now, if fluidity is present, lipid can show two types of movement. One is lateral, another one is flip-flop. What are these movements? How it takes place? I will explain later on. But just remember that fluidity, due to fluidity, lipid shows two types of movement: flip-flop and lateral. On the other hand, proteins are very large in size, so they cannot show flip-flop movement. They will only show lateral. Again, what are these movements? How it takes place? I will explain it later. This clear? Clear? Now, if something is fluid, then there should be something to make the whole membrane stable. Because if lipids is fluid, then it will always try to move. But there should be something which should make things stable within the membrane. Cholesterol does that. Cholesterol is the structure which is intercalated. Like if these are the lipid molecules, cholesterol is present in between. And cholesterol provides the stability to the membrane. How it provides? If we get to extreme cold temperature, then our membrane doesn't freeze. Why? Because cholesterol is present. If we go to extreme hot temperature, our membrane doesn't melt. Why? Because cholesterol.
So because cholesterol is very temperature resistant?
No. Cholesterol maintains the stability of the membrane. Okay. Cholesterol as a molecule holds the lipid together. And it doesn't hold tough that lipid don't be able to move. But it holds enough to provide the stability. Understood? This cholesterol is not present in case of plants and bacteria. They have some other different kind of sterol molecules, but not cholesterol. Cholesterol is restricted to animals. So this is a positive side of cholesterol other than causing heart attacks. Cholesterol is
I had a question
which causes heart attack. Okay. Question.
How does cholesterol make the body stable?
What was?
I mean, how does cholesterol make this thing stable, sir?
It holds the lipid
plasma membrane
molecule so that it holds the lipid molecule so that lipid won't separate or lipid won't crystallize. Okay, so it protects that.
So what does it do?
Now, first draw this. It holds the lipid molecule and prevent crystallization or separation of lipid molecules. First, draw this diagram, then you will write that. Oops. I guess every one of you know the term hydrophobic and hydrophilic. Na? Phobic means
Yes.
hating, philic means love. Now start writing about it.
So what does quasi-fluid nature refer to again? Quasi-fluid.
Fluid-like. Quasi-fluid means fluid-like nature.
Sir, what would happen if the lipids, I mean the fatty acid chains, react with water?
Fatty acid chain,
Reacts with water.
it will disrupt. The whole structure will disrupt. You have studied about missiles. How detergent works on dirt. It is the same function. Detergent does the same thing. Detergent have also these lipids molecule with fatty acid chain, and it forms a missile around the dirt molecule. And when water comes, the phospholipid attracts the water, the dirt molecule with water.
Okay, sir. Thank you,
Okay. And in general, if membrane reacts with the chain reacts with water, then it will disrupt. Another.
Yes, sir. What is intercalated?
Intercalated means it doesn't. If these are lipid molecules, cholesterol are intercalated.
Okay, so in between.
Yes, present in between the lipid molecules.
Okay, sir.
So provide stability to
The membrane. Another, you are still asking something.
Yes, sir.
Okay, done?
Sir,
Done?
provide stability to the membrane, right?
Yes. Absent in bacteria and plants. Plants have their own sterol molecules called as phytosterol, and bacteria have their own sterol molecules. It won't be cholesterol. Cholesterol is only restricted to animals. But they will also do the same job. Now, next structure which is associated is proteins. Now, the word or the line used by Singer and Nicholson for protein was that protein are present as icebergs in the sea of phospholipid. Okay, if this is a sea of phospholipid, protein are arranged as icebergs in the sea of phospholipid. Understood this point? Proteins are arranged as icebergs in the sea of phospholipid. Clear? Clear. Now, these proteins are present in a mosaic pattern. Mosaic pattern means spots. That's why they use the term fluid. They use the term fluid for lipids and use the term mosaic for. That's why they named the model as fluid mosaic model. Understood? So proteins were present as icebergs in the sea of phospholipid or in a mosaic pattern in the phospholipid. Okay, that is in spots, not symmetrical, not like present. If it is present with this, then it will be present on the opposite side. That's why the membrane is asymmetrical in nature. Why? Because proteins are not periodically present. Okay, okay. Plus one more point. Lipid has two faticity. Like it is hydrophobic also, it is hydrophilic also. That's why membrane is also called as amphipathic in nature. Amphi means two, amphibians, two habitat, amphipathic, two faticity. Hydrophobic also, hydrophilic also. Understood? Understood? That's why this membrane is called as amphipathic and asymmetrical in nature. Clear? Now next point. Protein can act as a structural molecule, it can act as an enzymatic molecule, and it can act as a channel molecule within the membrane. So same protein can have different function associated with the membrane. Understood? Protein can provide channel for the movement of solutes. Protein can act as an enzyme for enzymatic catalysis. Protein can act as a structural molecule of the material. Now, proteins are classified into two groups. How they are classified? If I make a membrane, then the protein can be present on the periphery of the membrane, called as peripheral protein. The protein can be partially or completely embedded within the membrane. Then it will be called as integral protein. Integral or intrinsic. Okay, and this one is peripheral or extrinsic. Okay, now these proteins are classified based on that majority of their part is embedded or not. But remember, this is a very generalized statement. They are actually classified based on ease of extraction. Ease of extraction means if a protein is easily extracted from the membrane, then it becomes peripheral. But if a protein is tough to extract, if a protein is that tough to extract that we need to disrupt the whole membrane to take out the protein, then it will be called as integral. Understood? Understood? Now, one question. This protein is intrinsic or extrinsic? Common sense says intrinsic. But this protein is actually a peripheral. That's why I told that according to our understanding, those proteins which are embedded are tough to extract, so they will be classified as an integral protein. But this protein, being very small in size, they are very easy to extract. So they are classified as peripheral. So the only basis for classifying protein into two groups was ease of extraction. How easily protein can be extracted. If we need to disrupt the complete membrane for extracting the protein, then it will be considered as a integral. Understood? Understood. Right. First point, icebergs in the sea of phosphorus.
So what do you mean by icebergs in the sea of phosphides?
Phospholipids.
Yes, sir.
Icebergs, like icebergs is present in the sea now. Same proteins are present in phospholipids.
So that's a reference to the in the cell membrane or what?
We are studying.
Okay, so in the cell membrane, you mean?
Of course. That's one example.
Sir,
Yes.
can you explain how proteins act as structural enzymatic molecules or present as channels?
If they are helping in the formation of membrane structure, then they are structural molecule. There are some protein which acts as enzyme, like there are number of proteins which are associated with plasma membrane of mitochondria, which acts as enzymes, and channels I will explain after some time in the functions of plasma. The protein which pass across the membrane are transported. Done everyone?
Yes, sir.
Okay. So,
Sir, in the diagram, what is the second type of peripheral protein?
this one,
Yes, sir.
that is also a peripheral protein because that is easy to extract. That is going across the membrane, but it is easy to extract. So it becomes a peripheral.
Okay, sir.
Okay. This same structure is given to NCERT. Okay. Now, now, the third part of membrane is carbohydrate. Carbohydrate is very small. What does carbohydrate do? Okay. That if I make a membrane, this is the lipid bilayer. On this, number of proteins can be present. Proteins can be present like this, it can be present like this, it can be present like this, it can be present like this. Okay. Now, there are carbohydrate chains which are associated with proteins or lipids towards the outer side of the membrane. Okay. What does these carbohydrate chain do? This carbohydrate chain help in cell-to-cell interaction. Example, RBC cells. RBC cells have antigens on their layer. These antigens, they have sugar molecules on their layer, from which RBC, if I have B blood group, then if I am infused with A blood group, then that A blood group RBC cells will have A antigen. So I will have antibody for A blood group. So my body will recognize the antigen of that A blood group because that is not my blood group. Understood? So the carbohydrate chain helps in cell-to-cell interaction. When a cell comes in contact with another cell, then both of these cells can recognize each of them or each other. Carbohydrate helps in that interaction.
So what's the name of the carbohydrate chain?
There is no specific name for that. Different cells have different carbohydrate chains. Okay, so there are different kinds of carbohydrate chain, and this helps in that chain, that cell-to-cell interaction. Understood? That's the only role of carbohydrate. But it plays a very important role because if one cell interacts with another cell, then it needs carbohydrate chain. Okay, so third point, yeah. As sugar chains.
So one more minute.
Those who are done, write the function of cell wall. Don't write it with cell membrane, mention it somewhere else. Okay. Mention it nearby cell wall, somewhere nearby cell wall, either on the header part where you have blank space in your copy. Okay. These are function of cell wall, don't mention it here. Mention it somewhere nearby cell wall. Mechanical support and maintains the shape of cell. Helping cell interaction. Prevents the input of molecules. Next, the cell of infection. Another.
So what are macromolecules again?
Macromolecules are those which are much larger in size as compared to the space present between cell wall molecules. Okay. Mostly they are polymers. Done? Everyone done?
Yes.
Okay,
Yes,
I
sir.
still have 5 minutes. I will make the diagram of cell wall. Just copy that diagram. Everything done.
Yes, sir.
It's a very generalized and simple structure of plasma membrane. In your NCERT, they have given a 3D structure of plasma membrane. So even if you will draw this diagram, still you will get marks. And use this diagram to draw in terms of Done? Everything well? Okay. One point in this, which I was telling, flip-flop movement and transverse and lateral movement. Listen, everyone carefully. If this lipid molecule comes on this leaflet, and if this lipid molecule goes to that leaflet, each of these lipid bilayer is made up of 2 leaflets. This is the first leaflet, or also called as the upper leaflet, and this one is the lower leaflet or cytoplasmic leaflet. Cytoplasmic or lower leaflet. Okay. So if the lipid molecule change the leaflet, then this type of movement is called as flip-flop movement. upside, downside, downside, upside. Okay, but
What do you call it?
flip-flop movement. Okay, but if a larger protein molecule, it cannot show a flip-flop movement now because it is very huge in size. So protein molecule can only move laterally. When it moves laterally, this type of movement is called as lateral.
Oh,
This
also,
lateral
sir,
movement
protein
can be
molecules are
shown
very
by the
rare
lipid
also, right?
molecules also. Okay, this type of movement can be shown by lipid molecules also. Understood? Any doubt? Any doubt?